All right, everybody, I'll be there in just a moment. I've got to go feed my dogs. I'll be there in a few minutes, no more. All right. Sorry about that. Didn't plan my morning very well. All right. So where were we? Anybody give me a refresher as to where we were in control of gene expression for eukaryotes?
The transcriptional regulation in eukaryotes slide.
Awesome. Thank you. So had we covered? So we finished up here, right?
I think so, yes. I don't know if there's like other stuff you were going to say about it.
Oh, no, that's cool. Just so I kind of figure out whereabouts I am in the order of things. My now fed dogs are avoiding the Roomba, which is kind of funny. Up on the couch like I am. Okay, so we talked about a lot about chromatin modifications. And just to recap, those broadly fall into chromatin remodeling. So where those histones are and, adding or removing them. Histone modifications themselves. So essentially acetylation and methylation of histones, which have, generally speaking, opposing, good morning, love, opposing actions, acetylation being pro-transcription, and methylation generally, though not always, being pro-suppression. And we also talked about methylation of cytosines, which is almost always suppressive. So, and also don't forget that those three things interact, right? Which is really important. It's what adds even more complexity. Okay, so once the DNA is actually available for expression, now we have the control of transcription, right? And that's really down to what DNA sequences are present in the regulatory promoter, which is, it's actually shown really small here, but it's very large and ill-defined, to be honest. It's really hard to figure out exactly where it ends, right? Because you have to know what DNA sequences are involved. And in addition to those DNA sequences, so that provides the binding sites for proteins that will either activate or suppress transcription. And so the specificity of gene expression, so where protein or where genes are expressed in response to what, essentially the whole kind of gamut of what, where, how much, and in response to what, depends on those DNA sequences where transcriptional activators and repressors bind, and what proteins are present in that cell. All right, and that's really kind of all of it, essentially. That's the whole thing. Now, one thing that will bend your minds a little bit is that these transcriptional activator proteins and repressors, they are also the subject of this control of gene expression. So these control expression of genes, other proteins control the expression of these, right? So now you can see how we can kind of canalize different tissues so that liver cells produce only particular proteins, muscle cells produce only particular ones, heart cells only particular ones, and so on and so forth, right? It's kind of like the ancestral record, in a sense, of where those cells came from. And so this core promoter, which is where the basal transcription apparatus assembles, so that's RNA polymerase, TBP, transcription factors, and so on, is around 500 base pairs, give or take, 400 to 500 base pairs. And that's often called the minimal promoter, because it's the bare, bless you, Said, the bare minimum required for transcription. Doesn't mean you're going to get much transcription from that promoter, but that's the bare minimum required for that gene. And the regulatory promoter is very hard to define. Can be thousands of base pairs and have regulatory regions, basically DNA sequences. Sorry, wrong key. Hundreds of kbp away. And we'll see an example of that in a little bit. And so all together, those DNA sequences, right, which are recognized by proteins, are called cis regulatory elements, or CREs, right? And they are all found in the regulatory promoter. Now, this is just like a laundry list. It's not 100% complete, but it's good enough for our purposes. And essentially, you have, kind of broadly speaking, three main types of DNA sequence, right? You have enhancers, and they're really defined by the outcome, right? Much the same as with bacterial gene expression, right? We have enhancers. Those are involved in enhancing gene expression through the binding of activator proteins. We have silencers, which, as you could guess, are involved in silencing gene expression. These are recognized by repressors. And we have insulators, and those are really all about walling off different genes. So you don't have accidental activation by an enhancer for another gene turning on a gene that shouldn't be activated in response to that protein. And so these are often called, particularly enhancers, but sometimes silencers too, response elements. And so response elements are how eukaryotes coordinate gene expression. And their DNA sequences recognized by a specific protein. And those sequences are found upstream of I don't know, genes involved in the same process. So in prokaryotes, they coordinate gene expression by simply lumping all of those genes together into one operon. So then you either turn it off and you get all of them, or you turn it off and you get none of them. Eukaryotes also need to coordinate gene expression, right? It's also important because for the same reasons, right? You know, you need all of those gene products present for a particular process to be successful. However, eukaryotes do it by having the same bit of DNA in each of those regulatory promoters. And so wherever that DNA is present, if the protein that recognizes it is also present, all of those genes will be turned on. So that's a way of switching on or not switching on a larger suite of proteins or genes, essentially. And these response elements are modular, or even the cis regulatory elements are modular too, right? Because they just require a short section of DNA. And so you can have multiples of those sequences. And actually, how many you have often determines how strong expression from that promoter is. So the one at the top is a SV40 is a simian virus, hence SV. And this is often used to make large amounts of protein. So if you want to express a protein for purification, say like insulin, I don't know why I keep coming back to insulin, but I do. Then you simply stick your gene of interest that you want to express downstream of an SV40 promoter, and you'll just get gangbusters amount of protein. Actually, so much protein that you can't use it for every gene you want to express, because that is often, that level of expression is sometimes toxic. Right, so lots of these GC boxes equals really high levels of expression, which makes sense because it's a virus, right? It wants to make lots of itself before the immune system comes along and shuts down the party. Now, these other genes have a different array of boxes. And so I'll give you a really cool example. Have I talked about butterfly wing spots with you lot before? Yeah?
You do.
Say again, Jesus?
Yes.
Oh, I have. Okay, so that's it. See, I can never remember what I talk about to one class or the other. I want to make sure I don't repeat myself and you lot get bored. So that's a great example of this modularity, right? And those boxes can be acquired. And that changes function quite significantly, right? Because function in terms of that protein expression is tied to what DNA sequences it has in its promoter. There are also, again, I'm sorry for doing this, but I got to check. Oh, I'm teaching, sweetheart. I think my daughter has asked me to open a bottle of something. Have I shown you my paper with the alternative promoters in it? No. Where are we at? So this was a gene that I worked on some years ago. Not really doing an awful lot on it anymore. But this is a gene involved in the protein is involved in recycling thing called nicotinamide, which is a byproduct of NAD metabolism. So NAD can be consumed. It's not just an electron carrier. It can be consumed and then it needs to be turned back into NAD. Otherwise, bad stuff happens. And so this gene Come on, scroll up a little bit, is fairly complex, right? It has 3 different promoters, it has 5 exons, and the active part of the protein is encoded by the last 3, right? And so these different promoters essentially, well, these 2 control in part where it is expressed. This exon has a signal sequence, which basically tells that protein to be exported out of the cell. So when this promoter fires, it produces a protein which is exported. It's an extracellular protein. When these 2 promoters fire, that protein functions only in the cell in which it's expressed. So you can get a very, very different outcome in terms of function, simply depending on which of these 3 promoters is active. Right. And when you actually look at that in terms of localization, right? So a big chunk of the work that I did, really big chunk actually, was sticking a green fluorescent protein, right? So this is from jellyfish, and it is fused. So it's translationally fused, right? It's actually part of the protein being produced. So wherever this protein is expressed, you will get green when you shine blue light on the worms. And you can actually do this while the worms are alive. It's actually really annoying because for some reason, worms are sensitive to blue light. So as soon as you turn the laser on, they start bloody moving. So you have to paralyze them pretty thoroughly. Otherwise you get crappy pictures. What do you want? Oh, that was a dog, not a child, just in case you were. And so when you do that, this is what you get, right? Actually, let's see if I can show you a little bit more closely. So this kind of stands in a little bit for our transgenic stuff, which we may not get to. So this is the secreted protein. This is the uterus of a worm. So you can actually see the embryos developing here. Here's a 1 cell, 2 cell, can't count cell, and lots of cells, right? And they pop out just here. This is the vulva. So this one, you can actually see it diffused in the space in between those eggs, right? These are matching kind of white light and fluorescence images, right? And so this tells you that this protein is secreted. It's floating around in that extracellular space. Now, when you put it individually under the GFP, under the individual promoters, you can see that you get very different gene expression. This is the head of the worm. It's actually got like a turkey baster, kind of squeezy bulb thing that sucks up bacteria. And wherever you get this white light, because that's actually what the camera records, we just color it green afterwards. You can see that one promoter is expressed heavily in neurons, right? And you can actually, if you zoom in a bit, and the picture's a bit fuzzy, you can see the dendrites, like the actual processes of those neurons. You can see a little bit better over in this image here. So there's a nucleus, the cell body of the neuron, and these are the neurites, sorry, the processes. However, when you look at a different promoter, now it's expressed largely in the intestine. So you get very different patterns of expression depending which promoter is active. And that presumably, we don't exactly know how, is tied into its function, right? For whatever reason, maybe the intestine is an active site of NAD consumption, so it produces a lot of nicotinamide, which is what this enzyme converts into something else. Right, so if there are alternative promoters, there's clearly a reason for that. Doop-a-doo. Now, next up on the list are enhancers. right? And so these are, again, regions of DNA which are recognized by proteins, and those proteins, transcriptional activator proteins, are typically required for transcription. So if they're not present, you don't get transcription. And so which ones are present drives what genes are expressed in which cells. So this is just a graphical comparison. And a thing to stress really, really strongly, every cell has the same DNA. Red blood cells accept it because they don't have DNA, right? Every cell has the same set of instructions. Liver cells are very different from lens cells, right? If you had liver cells in your eyeballs, you wouldn't be able to see what I'm doing, right? Because they'll be busy detoxifying, whatever nasties you've got going around your body, and not actually allowing you to see where you're going, right? So this would be really hard. You'd actually be listening to like a podcast instead. And vice versa, there's no point expressing the genes required for lens cell function in the liver because the liver's inside your body, don't need to do those things, right? And so these are two genes. I'm not entirely sure what albumin does. I know it's one of the main proteins in egg white, for what that's worth, but presumably something specific to liver function. Actually, let's look it up because that's just going to bug me. Oh, well, there you go. Helps keep fluid in your bloodstream. So it sounds like some kind of carrier protein involved in blood homeostasis. Anyway, so same genes in the liver as you have in the lens cell. The albumin genes expressed in the liver because the activator proteins specific for that gene are present. The ones for crystalline gene expression are not, right? And so in the lens cell, it's the opposite. The activators for albumin gene expression are not present because they're not expressed. And they're not expressed because the lineages and like the development of those two tissues are very, very different. So again, it's like this kind of memory of what is expressed dictates what is then expressed and so on and so forth to get you to those final tissues. And so instead you get crystalline gene expression. And crystalline is involved in clearing the contents from your lens cells. That's how you can see through your lenses, because they do consist of cells, right? Because everything in your body does. But those cells are transparent, which is how, I can see you and you can see me if you got your camera turned on. Right, so crystalline is involved in getting rid of the organelles and the various stuff in those cells so they become transparent, which is pretty cool. Silencers work the opposite, and they do so by essentially recruiting proteins that interfere with activator function or with RNA polymerase assembly. And so if those silencers are very close to the core promoter, let's see, put it back up. This one may be even better. So if those silencer regions are very close, proteins bound to them may physically obstruct the assembly of the basal transcription apparatus. Or they could do the same for the transcriptional activator proteins. So sometimes the sequence is very similar. So repressors will actually recognize a very similar sequence to a transcriptional activator protein. Sometimes the sequences overlap. So if one protein is bound, the other one cannot, right? And so this adds an extra level of control to gene expression. And just to give you I might have talked about Mr. Tuxedo before. Have you seen Mr. Tuxedo in his many paws already? I might have jumped ahead, I can't remember. Yeah. So the expression of that gene is controlled by an enhancer and a silencer, which are several 100,000 base pairs upstream of the sonic hedgehog gene, right? And if you interfere with either of those, either the making the enhancer work better or getting rid of the silencer, then you get ectopic expression here, which gives you, ta-da, Mr. Tuxedo and his amazing multiple toes. I have to share, I'm trying very hard not to look at more pictures of polydactyl kittens. Really hard. I'm going to drink some coffee so I can do something else, just to prevent myself from being tempted. Because they're so cute and they have huge paws. All right, okay. If I wasn't wearing a headset, I'll be slapping myself right now, just to make sure. Okay, and so the kind of the final part of those cis regulatory elements I want to talk about are insulators. And so the main deal of insulators is that they recruit proteins that prevent these enhancers from cross-talking. So these enhancers can work in either direction. Essentially what they do is they work by, some of them at least, bending DNA so that they allow those activated proteins to interact with the basal transcription apparatus. And so what insulator proteins do, and you'll often find them kind of, you know, in between genes, is that they physically prevent that DNA from bending. And so what that does is it means that this, if there's a transcriptional activator protein bound here, it can't bend the DNA over to the left and allow it to interact with the transcription apparatus in the promoter for gene A, and vice versa. Sometimes you might want that to actually happen, right? If you want to coordinate the expression of these two genes at the same time, that would be fine. But if you don't, you've got to stop that happening somehow. And that's what insulators and the insulator binding proteins do. It's essentially kind of walling off genes and their regulatory regions so that you don't accidentally activate another gene elsewhere in the chromosome. And as Mr. Tuxedo shows, these regions can be a really long way away, right? And so you really want to make sure, and there are going to be multiple genes in between this enhancer and sonic hedgehog, right? So you want to really make sure that anything that binds to that enhancer doesn't accidentally activate expression of a different gene. Because the specificity comes from the binding of the proteins and what proteins are present. Once those proteins are bound, they can activate transcription. Okay, anybody got any questions? Oh, hang on. Sorry, I just moved my chat over, Kiana. Yes, the insulator binding protein does prevent essentially this from happening, this DNA bending, right? Because you've got a gene here and it's core promoter, and the enhancer may be kind of over there, right? So the only way that protein bound here is going to interact with this gene is if the DNA bends over and brings that protein close to the basal transcription apparatus. Now, my arm doesn't bend the other way, but essentially if you had another one over here, that could also bend over and interact. So there has to be some way of preventing that from happening, right? And that's what the insulator binding proteins do, is they physically kind of clamp onto the DNA and make it more rigid. They stop it from kind of bending over. which is pretty cool if you think about it. Does that make sense?
Yes, it does. So if this bottom image were to have an insulator binding protein, it would be before the bottom transcriptional activator protein.
So if this enhancer was for a different gene, say one over to the left, then yes, there would be an enhancer between those two. Not enhancer, sorry, an insulator. You don't want an insulator between this enhancer and this gene because this is actually responsible for activating this particular gene. So it's not so much to prevent the DNA from bending between an enhancer and the gene it's supposed to activate, but to stop it from bending and enhancing a gene that it's not supposed to. So essentially the different genes are walled off. The regulatory promoters are still able to kind of bend and interact because otherwise these enhancers wouldn't be functional. But it stops it from accidentally activating a different gene. So you don't have insulators between all genes. It's not really that every single gene is walled off. It's just ones where you want to very much make sure that you don't get accidental activation of transcription. Does that make sense?
Yes, it does. Thank you.
Cool. And again, that kind of brings up a really good point. Not all of these cis-regulatory elements are present for every gene, right? So you may, obviously you're going to have enhancers because otherwise you can't activate expression. There may be no insulators or silencers present. There may be loads. There may be multiple different enhancers depending on, you know, the transcriptional outcome for that gene. Okay, so we've got a wee bit left to do. I'm going to try and get through this and leave a little bit of time left for looking at CRISPR-Cas9, simply because that is super, super cool. And if I check schedule, yeah, this is the last class, which is very sad. Because your last exam, not the final so much in the classical sense, but exam 4 is the Tuesday you come back from Thanksgiving. Which is kind of sad in a way, because it means you're going to be worrying about that over Thanksgiving. So you have to kind of compartmentalize, you know, eating lots of food and studying for your exam. So my apologies about that. It's just the weird ass way that this semester is organized. So let's crack on. So remember we talked about, Roomba has finished. Peace has returned. We talked about splicing, right? So removing of introns in eukaryotic DNA. And it has to happen because you only want to translate the exons, right? So you have to end up with a mature mRNA capped at both ends, one with a 5' cap, one with a poly A tail, and a continuous reading frame made up of those exons. So this happens to every eukaryotic mRNA. Every single one has at least one intron. Some of them have lots. Okay. Now, because we are removing bits of mRNA, now we can actually choose which bits we remove. So we always have to remove the introns. That's a given, right? You can't leave introns present, otherwise you'll get you'll get a nonsense protein produced, most likely a truncated one too. But now we can actually choose, well, not consciously, obviously, but we can change the protein produced from the same gene by leaving out an exon or two. And this is actually a super cool example, right? This doesn't happen for all eukaryotic genes. Actually, it doesn't happen for like a huge number of them either. It's somewhat rare, but it is a very cool mechanism.
Can you repeat the thing you said about if we leave out an exon or two, we can control what?
What protein is produced? And so the exons contain the information for the amino acid sequence, right? The polypeptide sequence. And so if you leave out an exon, right? So instead of essentially the splicing will be of the upper one, does that, right? So essentially the spliceosome will come here and here and it'll remove this intron and this intron. Does that make sense?
Yes, thank you.
Yeah. And so what you can also do is, and I think this is done by controlling which SNRPs are involved in the spliceosome, I think. You can do that instead. And so you're still removing these two introns, but now rather than cutting here once, twice here, now you just cut twice. Once at the beginning of the intron here, and once at the end of the intron just before exon 12. So you have exactly the same DNA, right? You have the same pre-mRNA produced. You have two different mRNAs produced. So you have two different proteins from the same DNA, right? Which is a pretty trippy kind of concept to think about. And this is actually a really cool example, right? If you kind of dive into this a little bit and give me a little bit of time to do so, you'll see how cool this is, right? So essentially what this is all about is a gene called the insulin receptor. So what slide is this? And the insulin receptor detects insulin levels, right? So, and there are multiple different insulin receptors that do different things in different contexts. But in this case, what this does, receptor detects insulin, right? And that's the kind of soluble hormone that's produced by your pancreas, your beta cells, when your blood sugar level goes up. When it detects that insulin, it tells the cell it's on, right? Because it's actually, if this is the cell, it's sticking out the surface, right? It's on the outside of the cell, right? That's how it can detect something outside. It tells the cell it's on to take up glucose. And it does so basically by making that cell put a glucose transporter into its membrane, right? And so you have two main tissues which take up glucose. I think the brain has got to be part of that, but I'm actually a little bit hazy on that, so I'm not going to talk about that. But the main ones are the muscle and the liver. Muscle makes a lot of sense, right? Because you're turning glucose into ATP so you can do this, right? Flap your hands around, or have your heart pump, or your intercostal muscles contract so you breathe, and so on and so forth. Right, so muscles are big consumers of glucose because they're big consumers of ATP. Pardon me. Your liver, on the other hand, it does use glucose, but its main role is to store glucose as glycogen. And glycogen is basically the animal equivalent of starch. right? So it's a storage molecule. And so if you think about it, you want glucose be taken up by the muscle cells first, right? Because they need it and they can't store it. So you really want muscle cells to take up glucose first. So how do you do that? Well, what you do is you make the muscle cells more sensitive to insulin, right? So as your blood sugar level starts rising above its upper limit, you're going to produce or release rather insulin from the pancreas, but it's not going to be crazy amounts, right? It's going to be a measured release. And so at the beginning, those muscle cells are going to be able to detect that because they have a higher affinity for insulin, right? And that's because exon 11 is removed. Presumably that changes the structure of the protein, so it's more likely to bind insulin, right? So when insulin levels are low or lower, low-ish, muscle cells are able to detect that insulin and take up glucose, right? Now, if glucose keeps increasing, which probably in my bloodstream it's doing right now because I'm digesting my breakfast, that's some mini wheats, right? So as my blood sugar keeps increasing, right, and it isn't coming back down because the muscle cells, even though they're taking up glucose, can't take up all of it, then you want your liver to kick in and take up the excess and store it as glycogen, right? So liver cells express a form of the insulin receptor which is less sensitive. It takes a lot more insulin present for it to activate. So you can get cell specificity in terms of how they respond to the same signal, not by expressing different genes, but simply making a different version of the same protein. And alternative splicing is one way of doing that. Alternative promoters is another way, which is what I showed you from that paper that I wrote a while back. Isn't that cool? I think that's super awesome. Love talking about this. Okay, next up is all about RNA stability. RNA stability and initiation of translation, which kind of goes a little bit hand in hand. So if we go back up to our guide all the way back up here, gone a long way, right? We've already talked about all of this, right? So we now have mRNA in the cytoplasm, and if there's mRNA in the cytoplasm, it will get translated. So if you want to control translation after transcription has already happened, now we're going to deal with either the stability of the mRNA. So if the mRNA is not there, it's not going to get translated. Or the initiation of translation, right? So there's two different ways in which we can do that. And generally speaking, that all centers around, dang it, where's that PowerPoint? The 3' UTR, the 3' untranslated region at the end of the gene. Right, so we talked a lot about this in terms of stability, in terms of protecting the mRNA. But this is also the target for either degradation or inhibition of translation too, right? And this is actually a super funky process. And I use this a whole lot in my own research. And essentially the idea here is we want to find those mRNAs that we want to shut down. How do you find an mRNA? You're going to express a sequence that matches it, right? And that's what this is all about. So this is called RNA silencing. Or as a technique, which I use in my lab, is called RNA interference, or RNAi. And it all centers around the expression of a non-coding gene. Right, so that's a gene that doesn't result in protein production, right, when it's transcribed. And so that results in a non-coding RNA that matches, in part at least, the 3' UTR of the target mRNA. I'm going to blow this up and shut off bits of it. Oops. So the very first step is that non-coding RNA has a sequence that allows it to fold and form double-stranded RNA. Now, double-stranded RNA is not something you find natively in a eukaryotic cell. It's actually something you find from a lot of viruses, right? Which is part of how this mechanism has evolved as a defense against viruses. You do obviously find, RNA secondary structures, right, in other places like tRNAs and stuff like that. But you don't find just small bits of double-stranded RNA on their own. And so when this is produced, now it's recognized by another enzyme. This is called DICER. And what it does is it chops up that double-stranded RNA into small chunks, about 20, 25 nucleotides long. Okay. All good so far? Okay, so this DICER enzyme isn't going to recognize like other non-coding genes like tRNAs or ribosomal RNAs and stuff like that. It will recognize specifically these small interfering RNAs or microRNAs. And it will chop them up. It needs to chop them up because the next part in the process is called the RNA-induced silencing complex, or RISC. And that recognizes and binds those small dsRNA fragments. DS for double-stranded. Here we are. And Argonaut is one of those proteins. Don't know why it's called that. Presumably someone liked watching Jason and the Argonauts when they were younger. So the next step, so again, this is double-stranded RNA, and it has to be double-stranded RNA because that's not kind of like a normal thing, essentially, in the cell, right? However, double-stranded RNA is never going to bind to another piece of RNA, is it? Because it's double-stranded. So then RISC, what it's going to do is it's going to get rid of one of those strands. That's called its passenger strand. So it destroys the passenger strand, and now that leaves just the guide strand. And essentially, I just get rid of that. Actually, let's do it one thing first. So it degrades the passenger strand, and now it's basically goes around with that bit of RNA in it and goes, is this going to match? Right, match, 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 match. Oh, found a match, right? And What the outcome of that is depends on how good of a match it is. So a near perfect match, and I think no more than 3 mismatches from memory. I haven't looked at this in a while. Between the guide RNA and the target mRNA equals risk cuts the mRNA. So what do you think is going to happen once that mRNA has been cut? So remember back to what we talked about in terms of RNA stability and the 5' and cap and the 3' poly A tail. What's the purpose of those 2 different structures, at least in terms of stability?
I have a question. So on part C.
Hey, you got to answer my question first.
Oh, I don't know.
Okay, all right. All right, then I'll let you off. Part C.
So for part C, after it gets the little strands of RNA, of mRNA, and then it goes
Oh,
to
no,
D-
no, not mRNA. mRNA encodes for proteins. It's coding RNA. What we're talking about is non-coding RNA. Have to be really specific about that.
Oh, okay. So the cut up pieces in part B are non-coding RNA.
Most definitely.
Okay. And then whenever it gets onto the RISC, the little like thing, is it coming to D to get cut up again? That little like picture kind of confuses me because.
Right. Oh, that's so this part here. Let's see if I can. So what this shows is that this is what it looks like once it's bound that small double-stranded fragment, right? That 25 or so base pair fragment. Now, before it can recognize a mRNA, so a coding RNA, it has to get rid of the passenger strand. So if you look at, don't worry about that top line here, but just look at this bit here. It only has a single strand of RNA present in it. And so it has to get rid of one of those 2 strands. Because if you think about it, you can only get double-stranded RNA, you can't get triple-stranded RNA, right? And so for that guide RNA to recognize its target, right, and its target might be, several thousand base pairs long, it has to be single-stranded, so it can actually base pair. Same as DNA base pairs, the same as mRNA DNA base pairs. We have to have single-stranded and single-stranded for those to come together.
Okay, thank you.
Yeah, so this is what it looks like first, and then it gets processed, so it just has 1 strand remaining. That's the guide RNA. And so back to the original question, what's going to happen if an mRNA is cut in between the 5' cap and the 3' poly A tail? What happens when there is a bare end, bare RNA end? What happens to that RNA?
It gets a cap?
It would if it was being transcribed, but this is way after transcription. So the capping machinery isn't going to work on this one. So what would happen if it doesn't have a cap? Sorry, what was that, Jesus?
RNAs?
Yes, exactly. And Jacqueline got that as well. So an RNAs will recognize that free cap end, right? Because there's no cap and there's no poly A binding proteins. And it will chew up that mRNA. So if the mRNA gets cut, RNases will recognize it and degrade the mRNA. Equals no more mRNA. And that shuts down translation completely. Because if there's no mRNA, well, then you don't get any translation. Now there's an extra little wrinkle which happens if that match isn't near perfect, right? So this is if there's a near perfect match. If there's a less than perfect match, and I can't remember the minimum number off the top of my head, then the risk and guide RNA bind, and instead of cutting the mRNA, the risk blocks initiation of translation. And it does so typically actually from preventing that mRNA from bending over and the 3' poly-A tail or binding proteins from interacting with the small ribosomal subunit. Which, if you, oh, where be that? Here we go. So you can see here that the poly-A binding proteins are actually involved in the initiation of translation. Can't remember if it's the completed ribosome or the small subunit, but it doesn't matter. Anyway, if RISC binds here, essentially it prevents the mRNA from bending round and translation does not begin. So essentially it's the same outcome. It's just less of a catastrophic outcome, right? So instead of degrading the mRNA, now we simply block translation instead, which is pretty cool. And so in my work, you can actually get bacterial clones that express double-stranded RNA that corresponds to every gene in C. elegans. And so all you have to do is feed those bacteria to the worms and get them to express that, and it knocks down expression of that target gene, which is a super, super, super cool technique to use. And actually won the people that discovered and developed it a Nobel Prize, which is one of 3 Nobel Prizes for work in C. elegans. It's almost guaranteed. Work on C. elegans, get a Nobel Prize. Super easy. All right, so to finish up, and I'm going to try and get to CRISPR-Cas9, really am, promise. Essentially, that's not it, right? So even after all of that, let's go back up here. What do you want, dog? Right. Even after we've produced that protein, it may not be functional. Because to be functional, many proteins have to either get to the right place. So if you remember that protein I talked about that I worked on, it has to be exported out of the cell, right? So in the cell, doesn't do anything or doesn't do what you want it to. Out the cell, it does. So it has to be sent to the right place. Many proteins have to have stuff added to them. So proteins on the surface of a cell are often glycosylated. So they have sugars added for various reasons. That's actually what you recognize in terms of antigens for bacteria, typically, are glycosylated sugars. And they're often cleaved. Insulin is a great example. Another gene that I worked on a bunch is another example. If they're not cleaved, they don't work, right? And so all of those things have to happen to actually have active protein. We could talk about degradation of protein, but that's a whole nother subject. And there's no way we're going to have time to do that. So I'm just going to give you two quick examples, right? And so the first one is about something called phosphorylation. So phosphorylation equals the addition of a phosphate group to a protein, right? And you can add those to 3 different amino acid residues. I'm not going to get into which ones right now because it's not important for the understanding of this. And so this is work that a friend of mine did, a lady called Michelle Castellito, who's at UCLA, I think, still. And so this is a transcription factor, DAF16. So these are worms, as you probably guessed. And it's a transcription factor. So transcription factors or transcriptional activated proteins only work if they're in the nucleus. Makes sense, right? Because that's where the DNA is. And so to affect whether or not it functions, you could control whether or not it's produced, right? But that's not how it works for this one. You control where it is. And so if the transcription factor is out in the cytoplasm, right? So these are, actually, I'm not even sure what cells these are. Looks like they're in the tail of a worm, right? So if the protein is out in the cytoplasm, no transcription, right? If it's in the nucleus, which is what you see over here, and these again are green fluorescent protein reporter constructs. If it's in the nucleus, now you can have transcription. And that's done by phosphorylation. So if this protein has, and this is actually involved in insulin signaling as well, always comes back to insulin. If this protein has a phosphate group added to it, it will stay in the cytoplasm. You remove that phosphate group, dephosphorylation, by a enzyme called a phosphatase. Now that protein is free to move into the nucleus. And you can actually force one versus the other simply by changing the target amino acid. And it doesn't necessarily have to happen in that direction. For other proteins, it may happen in the opposite direction. But either way, what we're doing is we're not changing how much protein is present. We're changing the protein itself to change where it is active. And phosphorylation is one of the main ways in which that is done. It's not the only way, but it's one of the main ways in which that's done. And this is some really, really cool work that was carried out in a parasitic nematode. So this is C. elegans, and this is Strongyloides stercoralis, which is a parasite of humans and gerbils, funnily enough. Poor gerbils. Okay, so that's one example, phosphorylation. Another one, and this is really cool. So actually, oh man, I lost some more blood yesterday. I'm always losing blood from various things. So that one was working on my wife's Christmas present, the metal chicken. And if I had time, I'd show you it, but unfortunately I don't. So if you cut yourself, you want to stop bleeding, right? Because 2 reasons. One is really obvious, right? If you lose enough blood, you die. And the second reason is this is essentially an entry point into my body for things I don't want in my body, right? So I want to seal up this hole as quickly as possible so that various nasties that was probably on the whatever it was that cut me don't get in and cause an infection. So we have a situation where normally, i.e. when I'm not in the garage, I don't tend to bleed very much, right? And so I don't want this process to work. But when I do puncture a blood vessel of some sort, I want to seal that up as quickly as possible, right? That's not, so actually there's two different things going on my finger here. One is an old one and one is one I got yesterday, right? The older one has already started forming, tissue, scar tissue, essentially, right? That's way after the initial insult. Straight away, you just want to block the hole, right? And that's done in a extraordinarily carefully controlled process involving a bunch of different proteins. And so the two that I'm going to talk about are called thrombin and fibrin, right? And so this is a multi-step process. And again, you don't want this to happen inadvertently, because if it does get activated just in a normal situation, you will block a blood vessel. Because what we're going to do is we're going to make a protein change its properties so it sticks together. And in doing so, it sticks red blood cells together and it basically forms a plug called a blood clot. Now, you've heard of blood clots in terms of strokes and heart attacks, right? You don't want chunks of stuff floating around in your bloodstream because it is bad, right? But when you have a hole in one of your blood vessels, you want to fill it. And so this is a two-step process. And actually these clotting factors, lack of these is what's behind hemophilia. Right, so hemophiliacs don't produce, I can't remember which one it is, particular clotting factor, and so they cannot activate this cascade. And so if they have an injury to one of their blood vessels, blood just keeps coming out of it. And so if you're a hemophiliac, you can literally bleed to death from cutting your finger, which is just an astonishing kind of thing to contemplate. And so what happens here is these clotting factors will cut this inactive protein here called prothrombin. It's a bit of a mouthful. So when prothrombin gets cut and it becomes thrombin, now it too becomes active. And its job is to cut fibrinogen. And when it does so, that fibrin will stick together and it will form a mat of blood cells and fibrin, pardon me, that will block the hole in your blood vessel. And if you look at this in a bit more detail, so this is what, hang on, let's see if I can, there you go. This is prothrombin here, and oops, as it stands, this is not active, right? It's an inactive enzyme. However, when it gets cut, and it gets cut just around here, then you get your active thrombin. Right. And what that does is it removes these little bits labeled A and B. There are actually A and B on the protein, but they're called parts A and B. And that allows those fibrin molecules to stick together. If those are in place, those fibrinogen molecules just float on past each other, right? They're not sticky because those A and B parts keep them apart. It kind of, it's like a sticky bit on a Band-Aid, right? It's all covered up. But when thrombin cuts them off, now these form, and they form these huge complexes of cross-linked fibrin. And those trap red blood cells and platelets, and that together blocks your blood vessel, or the hole in your blood vessel. And that basically just plugs the leak until there's time enough for the tissue to grow back around it. So the next time you you know, cut yourself, or even if you give like a blood draw or something like that, remember this because it's really, really cool. That's essentially what keeps you alive after this. But it's also something that could kill you very quickly if it goes wrong. Right. And so this multi-step process makes it very, very tightly controlled. Now, just quickly, because I promised, and I only got a few minutes left, which isn't really enough time to do it justice, but I really just want to talk about this because this is super cool. So you got fluorescent bunnies, very popular at Christmas by all accounts. But what I want to talk about is CRISPR. Oh, there it is. I knew it was around here somewhere. There you go. Yeah, I knew I put it in a PowerPoint somewhere. There's the link. Right, I'm not going to talk about RNA. We're going to talk about CRISPR-Cas9. And so I actually had a friend who worked on this, not as a technique, but as a system. So essentially, this is a really, really cool thing. It's essentially one of the scariest things in biology, because this allows us to make any genome edits we want. And we can do that in humans. It has actually been done in humans. Two girls in China were very unethically genetically modified by a Chinese scientist to be resistant to AIDS, to HIV, sorry, not AIDS. So the cat is out of the bag. Pandora's box has been opened. We can actually genetically modify humans, which is just terrifying. But this has actually much, much more humble beginnings. Essentially what this is the way in which bacterial cells acquire immunity against viruses. And so it's kind of funny to think about bacteria having an immune system, but they do, right? And so if a bacteria is infected by a virus and not killed by it, it grabs a little bit of the viral DNA and sticks it into a cassette. That's part of what CRISPR stands for. Can't remember. Something palindromic repeats. And so if it's ever infected by that virus again, what it does is it produces a little bit of RNA, right? That's called a guide RNA, or a single guide RNA. It's this. And what that does is similar to microRNA or siRNA that we were talking about earlier, for RNA interference. That recognizes that bit of viral DNA, and it directs this protein Cas9 to cut it. So it cuts that DNA, leads to that DNA being destroyed. Now, some very, very clever people saw that and thought, ooh, that's pretty cool. I wonder if we can use that to make genetic modifications where we want. And so now what you can do, and a friend of mine does this down in Houston, another A&M campus, is you can make that guide RNA to match your target DNA sequence, right? And so if you express that guide RNA and you provide this Cas9 protein, that's why it's called CRISPR-Cas9, what happens is that guide RNA directs the Cas9 protein to that bit of DNA, and it will cut it. It will cause a double-stranded break. And as we've learned, double-stranded breaks are pretty terrible, right? And typically, the response is a, oh shit, kind of response by the cell, and it will just kind of jam them back together again. And you often get a mutation as a result. So if you just do this alone, you will direct a mutation to that point. You're not going to know exactly what that mutation will be, but you'll get a mutation at that point due to that double-stranded DNA repair. Now, if you provide another bit of DNA, this is where it gets really crazy. And that's your donor DNA. All you have to do is make sure that donor DNA matches either side of that break. And so this essentially acts as a homologous chromosome for homologous recombination. So if you remember us talking about homologous chromosome, you basically use that other bit of DNA to fix that break. And in doing so, in this system, that other bit of DNA gets flipped into the chromosome. So you can put a GFP protein into a chromosome. You can replace that DNA with a bit of mutated DNA, mutated in exactly the way you want. You can do pretty much whatever you want. And so this is arguably the most powerful thing to come out of biology in the last 10, 20 years. It's also the most terrifying as well. Because now all you need to do is know what change you want to make. Say you want to make super muscled humans. You can actually get super muscled cows. Actually, they look ridiculous. It's like Arnold Schwarzenegger cows, right? They're called double muscling or Belgian something or other. Can't remember what. But now you can actually do that for humans. So you could make a baby, whether it wants to be or not, into a massively muscled baby. Jesus, PAM is a short, it's not a spray. It's actually a short sequence that is required for that Cas9 protein to be able to bind. I don't actually understand fully how it works, but there has to be that sequence near to where you want that guide RNA to bind for the whole thing to work. But it's just a very short, I think it's a trinucleotide repeat, and it's all over eukaryotic genomes. So yeah, there you go. I've gone over a little bit, but I thought it was worth the extra. So yeah, and that's it. We are done. Last exam or last thing is next Tuesday, and that will be your last exam. So yeah, it's kind of a bit weird, right? Because we haven't finished the semester yet, but we finished the semester. So same format, I'll be online during that class period, at least for the first like 45 minutes or so. If you have any questions, you can even use that to ask questions about this material if stuff comes up that you want clarifying. Because the exam will be open from 9:30 until midnight that Tuesday, which is Tuesday the, is that the 2nd? 7th, sorry. Tuesday the 7th. So yeah, same format as before, same number of questions, similar level of difficulty.
Sorry, you said our last exam is open from the 30th to the 7th?
No, from, I got the dates wrong. It's open from 9:30 the morning of the 7th to midnight of the 7th of December. That's exam 4. And I'll be there from 9:30 at least until 10:15 or so, probably just reading my emails and reading the news and stuff. If you have any questions, clarifications, stuff like that, I'll be there. Cool. All right, everybody, have a fabulous Thanksgiving. Try not to eat too much. Enjoy yourself and study hard at the same time. And I may see you in lab next week. Or if I don't, have a lovely rest of the semester and Christmas. All right. See you all later. Happy Thanksgiving to you too.
The last exam is just over 17 and 18, right?
Yeah,
like
and that little bit on CRISPR-Cas9 that we talked about.
Perfect. Thank you.
You're welcome. All right. Any other questions, just shoot me an email, okay?